Welcome everyone. I am Dr. Gausia Bashri from the Department of Botany, Aligarh Muslim University. Today we will discussing field and herbarium techniques and essential skills for studying, preserving and documenting plant species. And this presentation will cover the purpose of herbaria, steps in herbarium specimen preparation and specific techniques for handling different types of plants. So, let's discuss the herbarium techniques for which that we needed for the preparation of herbarium. So, first of all, previously as we have discussed about the herbarium, so just a quick revision, what is a herbarium? As we have discussed earlier that a herbarium is essentially a collection of dried plant specimen mounted on a sheet, often referred as a plant library. It serves as a bank of plant specimens and a reference for plant identification and storehouse for long-term documentation. So, we will discuss the specific techniques for collecting, preserving and documenting the plant specimens in today's lecture. So, moving forward to the techniques of herbarium preparation. Preparing herbarium specimen requires several steps to ensure durability and accurate representation. And the process includes collection, pressing, drying, poisoning, mounting, stitching, labeling and storage. Each step has a specific guideline that contribute to creating a high quality specimen. So, let's explore each step in more detail, and today we will going to discuss about in details in the collection, because this one is the most crucial step, because what we are collecting it's very important, because all our other process depending upon our collected specimen. So, this one is the most crucial process, that is the collection, and for the collection we have to visit the different places. That's why the topic of this today's presentation is the field and herbarium techniques, because we have to visit different places for the collecting our plant material. So, now start with the collection. So, collection of the plant specimen should bear flower, fruit if present. So, it is a just process of collecting a plant specimens with its reproductive structure, and it should be collected with roots or other underground plants, because we have to identify the different plants. So, all the plant parts are very essential during the collection, and a twig about 25 centimeter with leaves and flower will form an ideal material for the collection, because we have to prepare a herbarium of this particular collected specimen. So, it is required about 25 centimeter in length, because we have to place in a herbarium sheet which is a quite specific in size. So, we have to then note the sheet number and data recorded in the field notebook. So, the collected specimen we have to label the specimen. Then soon after the specimen collected, they should be pressed itself, which is the next step in the collection after the collection of specimens. So, what are the objectives of collecting a specimen? So, the main objective of plant collection goes beyond merely obtaining a specimen. It is about gathering comprehensive data, like common goals includes the documenting flora, assessing its diversity, studying rare species and collecting material for research or ethnobotanical studies. So, every collection outing should have a clear purpose and well planned. So, we have to plan before going for the collection. It is a very important for the collection of very well planned trip, because we cannot go to outside and just took any material and come back. It also requires money, so we have to a very well planned trip before going to the collection. So, these are the some objectives of the collection of herbarium material, like for the building of a new herbaria, enriching our old herbaria. As we have discussed earlier that the herbarium has a very significance in plants taxonomy as well as in the biodiversity conservation, for the ecological establishment and sustainability. So, it is a very important for the documenting a plant species. Then collecting plant material for writing a flora. Suppose we write to the specific flora of particular region, then we also have to collect the plant specimen. Then collecting live materials for the introduction in a garden or herbarium. Suppose we are going a particular area, those plants are not growing in our region. So we are collecting for that particular gardens, that is our living places. Then collecting material for particular genus or species for the research work or for a particular analysis. Then we have to collecting a large quantities for trade, then ethnobotanical study. These are some objectives of collecting plants. So depending upon our objectives, we have to plan our trip. So how much of the plant is required for the collecting? So now discuss what are the steps in the collection of the plants. So as I have told you, planning a field collection, then we have to prepare a proper planning is very crucial. Decide on the study area, gather local knowledge, consider environmental factor and secure the necessary funding for transportation and equipment. And selecting a team for motivating individual is also important, as field work can be physically and mentally demanding. So it is a very important to a very well planned trip for the collection of the plant material. Now moving towards the next step, that is the gather general information and customs of the area like literature, travelogue, maps and topo shoot, herbarium labels, laws and custom, accessibility, available support and climate. These are the some information we have to gather before going to that particular area we are going to visit. Then as I have told you that selecting a team is very important, because our morale may be quite be down during the process of a selection. Then the next thing is very important for the collection of plant, that is the equipment. Here are the list of the essential equipment for the field and herbarium preparation. Here is the list of essential equipments for field work and herbarium preparation. Like common tool includes the cutter, pruners, polythene bags, drying sheets, a field notebook, GPS and first aid box. Each tool plays a role in efficiently collecting and preserving specimens in the field. As you can see in these images, there are a number of tools which are necessary for the collection of plant material. Among them, some are listed here in detail. Like the first one is the vasculum. Vasculum is a metallic case of a tin or aluminum with a tight lid. It should be light in weight so that it easily carried to the field. Generally, the size of vasculum is 50 into 30 into 15 centimeter in the area, and the vasculum is useful for keeping the plant immediately after the collection. In the absence of vasculum, polythene bags can be served with the purpose. As you can see in this particular image, this one is the vasculum, and we carry the vasculum with the help of this particular handle. Because vasculum is important, because when we collecting the plant, we have to place it in the vasculum for the security, because if it is in an open state, it can be damaged. Then the next most important thing in the collection site is the field or plant press. It is made up of two hardboards or wireframes or thin plywood of 30 into 40 centimeter and tied by two leather or cotton straps. So this is also an important part for the collection of the plant material. Then the next important thing is the field book. Field book is also very important material for the collection of the plants, because we have to record some important data in the field book. And this particular field book has a very important labels, like you can see this one is the field book of a Botanical Survey of India, which have a different number of entries which have to enter during the collection of the plant. Like this is mean for the recording of a notes of the collected plants, because we cannot remember each and everything of each collected plant. So we have to note down in this particular field notebook, which have a different number of pages. So every collected plants we have to record them on the field book, and these pages of field books are especially printed in a book form, and generally they have 100 serial numbers. So just suppose in this particular field book, suppose it has a number 37. So the plant which is collected and the entries which have filled upon this particular field book, so just we have to label that particular plant with the number 37. So just when we back from our trip, so we have to match the characteristic of that particular plant. So that plant has a label 37, just we have to move down on the field book on the 37 sheet number. So we can get a exact information of that particular plant, because in the field trip we have to collect a number of plants. So we cannot remember all the entries for that particular plant species. So in this way we can easily prepare a document in the field. Then the next equipment is the collecting a pick for the dig. Then the next equipments are a collecting pick for digging up roots and rhizome of herbaceous plant, so that the collecting specimen is complete, like digger or towel. Then a strong pruning knife is also something required for cutting branches and other plant parts. Then a pair of pruning shears or skatiers for cutting woody and hard materials. Then like other equipments are blotting papers, collecting bags, hand lens, pencil, camera, altimeter. These are the some other things. And as we have discussed earlier, that field notebook, which is also very important. Then specimens have two parts, as I have discussed earlier, that the plant material and label, as label is an important part of the herbarium, because we are entering the data on the field notebook and we just putting the label on the plant material, so that we can remember which one is the entries for that particular plant. As you can see in this particular image, these are the some collecting equipments like hand lens. Suppose we have to identify a particular plant species, or we have to mention some important character of that particular plant in our field notebook. If it has a particular minute structure, then we have to also have a hand lens. Then these are the bags, that is also essential for the collection. Then we have some leak proof plastic bottles for the collection of aquatic materials. Then these are the field labels or diaries, and these are the silica gel to dry samples quickly if required for the DNA studies. So these are the some collecting equipments. These are the further carried over here, like this one is the plant press. As you can see, these are the made up of a two cupboard sheet. Then these are the scatia, these are the other equipments which are necessary for the collection of the plant material, as you can see in these particular images. When we collecting the plant, it's important to represent the population diversity without over collecting. So avoid rare species or those with only one or two individuals. So the aim of the collect specimen with flower and fruit, and these parts are often vital for identification. So we have to choose the plant that represent the species well, ensuring they include root, stem, flower and fruit, and document the collection with detailed notes including date, location, habitat and unique plant characteristics. And this information will be vital during the identification and future studies when we back to our original location from the trip. Then collect a specimen in dry condition, because a good time being mid-morning after the dew it is important to collect the specimens in dry condition. A good time being mid-morning after the dew has dried, but before the heat of the day causes the plant to wilt. So if specimens are at all wet, you need to wash soil off, then root, then dry them carefully before pressing. And use a pencil for these notes rather than pen, because any damp or wetness can cause ink or to submerge, and that is the unbearable. Then taking photograph is also a important step, because photograph is for the identification is very important. So take color pictures of each plant in a natural environment to show the habitat and close up of flowers and fruits. That is very important for the identification of the plant. Then use for this purpose, use a 35 mm single lens reflex digital camera, that is the DSLR, with a standard lens and tele and macro lenses, and also a tripod for better output. Then photograph taken should be recorded appropriately in the notebook, and the care must be taken to protect the camera from the rough handling and becoming wet. As you can see, this is the photograph of the impatient balsamium, and this is the photograph of the habitat, that is the alpine meadow. So we have to taking care of the habitat, and the close-ups of the photograph, as you can see, that clearly telling the habits and habitat of that particular plant. So besides this, we have to also making the clear observation, like observe the field, topography, vegetation and soil type. For each plants collected, document characters like height, flower, color and aroma, as these may not be preserved well in a dried specimen. So this observation is very important, because when the specimen become dry, these characteristics will now become not present in the dried specimens. So we have to document these characteristics before after the collection of that particular plants, and these observations are very important to note down in our notebook. Then, as I have discussed earlier, that the field notebook is very important, and a field notebook is essential for recording detailed information on each specimen and including collection number, location, altitude, habitat and physical description. And the proper labeling during collection helps to maintain organize a record in crucial and for accurate herbarium documentation. And if we see a typical field notes, you can see here it include the following information, as you can see in these particular images. Like the first, that is the collection number, or that is also called as the field number. Then name of the plant, like family, genus or species, what we know about that particular plant. Then province, that from where we collected, that is the state, district and country. Then locality, as a precise as possible. Then altitude, that's why the GPS is necessary during the field trips, because GPS tells about the exact altitude of that particular location. Then habitat, like type of terrain and some indication of the communities such as grasslands, pink oaks or forest. Then a description, like a description of the plant is always useful, like especially regarding the character that is not kept on the herbarium aspect, such as the height of a plant habit or flower color and smell. Then date of collection, and the lastly is the collector's name. These are the some important field notes that we have to note down in our field notebook, which is very important for the identification of the particular plant in later. Now moving towards the next thing, that is the what not to collect during our trip of the field trip. So we do not collect a plant or in areas with it prohibited without express permission of the designated authority. Then do not collect a plant if there is only one or few samples. Search for better population. Suppose we are going to visit a particular field where only 2-3 members of particular species existed. So we don't have to collect from that particular area. We have to move on some other areas where the population of that particular species is larger. Then do not collect a depopulated form, and not collect sterile samples unless sterile samples form are available, and continue search, mark the locality for the repeat visit. Then do not collect a particular species that repeatedly at a different location of the study area. Instead, make notes of its distribution in the field notebook appropriately. Suppose we have a particular area of a visit in that particular our field visit, and that is one species is existed in a many particular location. So we have to just note down from where that particular species is present. So that is tells about the distribution, and we have to collect only from a one single place, not from the repeatedly from again from the different places. Then the next step is the size of the plant specimen which we have to collect. That is also very important, because we have to paste that particular specimen in a herbarium sheet. So the plant specimen size is very important. As we know, the herbarium sheet have a standard sizes that vary slightly across the regions. Like generally the herbarium sheet size is the 42 into 28 centimeter in India, and 42 into 29 centimeter in USA, and 42 into 26 centimeter at Q, and certain other European herbaria has a 44 into 29 centimeter as in case of Natural History Museum in London. So these are the sizes of the herbarium. As you can see here, in India it has a 42 into 28 centimeter. So we have to collect a plant specimen that has the appropriate size according to this particular sheet. So generally, as I have told you earlier, that a twig of a 25 centimeter is the best for the herbaria preparation, as we have discussed earlier. So this is a perfect size, and in case of herbs, the whole plant with roots or underground parts are we have to collect because they have a very small size. So in case of a woody plants, we have to just collect a twig with around 25 centimeter in length, and for in case of a herbaceous plant, we have to collect a whole plant of a small size with all the parts from the root system to the shoot system. In shoot system, both the part vegetative as well as the reproductive parts we have to collect. So next, now moving towards the collection of a different types of plant. As we have discussed earlier, that the collection is a very important, and for every type of plant there is a different collection procedures. Like in case of aquatic plants, this collection procedure is very challenging due to their fragility and tendency to close when removed from the water, and use mounting chute underwater and gradually drain the water to spread the specimen naturally, which is very important in case of a water or aquatic plants, because suppose if we removing the plants from the water, its original form is get disturbed. So we have to place our mounting sheet inside the water bodies, and on that particular sheet we just have to overlay the plant, and then we have to cut that particular from the size. Then it has in a natural shape, and after that a piece of a wax paper over the plant top will prevent it from the adhering to the drying paper. Then for the tuberous parts, if any, should be scooped out and flattened to dry them easily. Dry them are a specific requirement for this type of plants. Next, in the aquatic plants, like in case of Limna and Wolffia, these are the very microscopic plants, and these plants should be collected in a mass, sun-dried and put in a packet, knotting on the sheets, and the packet should be pasted on the mounting board. So we have not to collect some important piece of a plant, just we have to collect in a pockets in a packet, then we have to dry them, and then we have to paste that packet in a herbarium sheet. And in case of inflorescence of typha and quercumum, often shatter and may be held intact by enclosing them in a bags. So bulky fruits and cones may be stored in a special boxes or cases, and a specimen should be clearly indicate that the both sheets and necessary material are present in the collection. So for these type of aquatic plants, there is some different collecting procedures. Similarly, the collecting procedures for aroids are also different. Like aroid have a thick epidermis and require a proper treatment to prevent regrowth in the press. So use high temperature or a solution like alcohol and formalin to kill the tissue. This sticky nature of some specimen may necessitate the muslin coat to prevent them from adhering to the blotting sheets. Then the next in the collection is the bamboos. Bamboos collection are rare and complex to their sterile state and large size. So detailed notes and extra equipment like gloves, saws and special markers are necessary, and the pictorial formats are recommended for the recording details quickly and accurately in case of a bamboo plant because of their size and sterile nature. Then in the collection of orchids, orchids also need a special preservation techniques due to their delicate structure, like boil the specimens briefly to prevent the leaf drop, and use alcohol formaldehyde to preserve the flower shape. And orchids are often collected alive for garden cultivation, so proper packaging and aeration are also essential in case of a collection of the orchids plants. Then next is the collection of a cacti. Cacti are preserved by cutting at joints and drying with the artificial heat. So note the arrangement of our spines and joints, as these are often diagnostic feature in case of a cacti species. Then for the collection of palms, palms are difficult to collect due to their height. So collection should include the leaf tip, rachis and the leaf sheath. If cutting the leaf, ensure the hustula remains intact for the accurate identification in case of palms. Then for the ferns, we have to collect them with the rhizome, as hair and scales on the rhizome are key for the identification. And large plants should be partially collected with exact measurements noted in the field book. So these are some collection tips for the cacti, palm and ferns. Then the next one is the collection of medicinal plants. When collecting medicinal plants, aim to gather reproductive parts like roots, seeds in large quantities for chemical testing, because we can get the characteristic of medicinal from these particular parts. And these tests determine which plant should be cultivated or collected further for the medicinal use. As you can see, this is one of the digitalis and this one is the Panax plant, which has a medicinal properties. Then to preserve the color, use a solution containing glycerin, alcohol and other chemicals. And regularly changing paper sheets and using salicylic acid treated sheets can help to maintain the vibrant colors during the drying process. As you can see, these are some solutions which we can use for the preserving the colors, like this one is the 50% alcohol or formaldehyde, glycerin, then glacial acetic acid, that is the 2.5 concentration, then cupric chloride and uranium nitride. So these are some processes which is important for the preserving the colors of the leaves and flower. Then in the last, there are some important points for collecting a good plant specimen. So generally whole plants with roots or any other underground plants are to be taken for the plant samples. But in case of a larger trees or plant having a height more than 4 feet, the twig with leaves and fruit and flower are to be taken, as we have discussed earlier, and it should be necessary that the plant sample have leaves and fruit and flower. Then in some plant species, the male and female plants are different. In that case, the flower must be necessary. Then the next is that if possible, you should select the mature plant parts, because morphology of young plant parts are maybe different than that of the mature ones. So it will cause a wrong identification. So it is a better way to collect a mature plant besides younger one. And to cut the plant, you should use a blade or cutter, not a hand, because it can damage the plant. And while collecting the creepers, you should ensure that the other plant parts are not taken with them. Then the last, if possible, take photograph showing the plant clearly of the plants and its natural habitat, as we have discussed earlier. So these are some important guidelines for the collecting the plant specimens. That's all for today's session, that we have discussed about the what are the important steps in the collecting of plants, and we have a very well planned trip before going to collect the plant specimens. So that's all for today's session. Thank you for joining me. Thank you all.